We all know what pain is, and we all have suffered from it, except for those people who suffer from Riley-Day syndrome. But do we really understand how pain works and what it actually is? The body is equipped with pain detectors in most areas of our body. These pain detectors are actually specialized nerve cells. These nerve cells are called nociceptors. Like all nerve cells, they conduct electrical signals sending information from all parts of our body to our brain. But these nociceptors only alert if something could cause damage to our body. In a simplified way, pain is our body's earliest warning system and protects us from the outer world. For example, if we gently touch the tip of a needle, we won't feel any pain. But if we push harder against the needle, we get closer to the nociceptor's threshold, and they fire as a signal to your body to stop doing whatever is causing that damage. The same happens with the fire. As we get closer to the flame, the threshold can be exceeded and we start to burn. This threshold is not set in a quantity, and some chemicals as ibuprofen can turn nociceptors, increasing the threshold for pain. When cells are damaged, they start producing chemicals that lower the threshold. Here is where painkillers do their job, blocking the production of these chemicals called prostaglandins.
Before we get into prostaglandin synthesis, let's start from the initiation of its precursors. Here we have a cell membrane which consists of phospholipid bilayer. If you focus on a single phospholipid, we can see that it has a hydrophobic head as well as a lipophilic tail. Using a special enzyme called phospholipase A2, we can liberate and convert phospholipids into arachidonic acid. In the following image, we have a picture of an arachidonic acid, which consists of 20 carbons and is a polyunsaturated fatty acid. Since it has more than one double bond, in this case we have 4. Fun fact, it is an omega-6 fatty acid, since its first double bond from the methyl site lie on the 6 subsequent carbon. That means it's also obtained from our diet. Now that we know more about arachidonic acid structure, we can review its function and what makes it a special fatty acid. Well, most importantly, it is a main precursor for arachidonics, which are a group of hormones that act locally and have a short half-life. We have 3 main types of arachidonics. The first type are prostaglandin, which cause inflammation, pain, and fevers. The second type of icosanoids are thromboxane, whose function are to form blood clots and reduce blood flow. Finally, the last kind of icodix are leukotrienes. Those function is to induce contraction of smooth muscles in airways. Since ibuprofen as well as other NSAIDs, non-steroid anti-inflammatory drugs, deal mostly with inflammation, fever, or blood clotting, we will be ignoring leukotrienes.
Now we finally have arachidonic acid extracted from the cell membrane. So, what is it used for? Well, the following arachidonic acid is a substrate for 2 important enzymes, one called cyclooxygenesis, or COX for short, and another called 5-lipooxygenesis. However, we're going to be focusing on cyclooxygenesis since it's the enzyme that's the precursor for prostaglandin H2, which is what causes inflammation, fever, and pain. which is precisely what ibuprofen can control. Cyclooxygenase can be expressed in two forms, which are COX-1 and COX-2. We'll touch back on these enzymes in a second. For now, we'll go back to our prostaglandin synthesis. Finally, arachidonic acid reacts with COX enzyme to form PGH2 or prostaglandin H2. The 2 in the nomenclature represents the number of double bonds. As we know, arachidonic acid has 4 double bonds, and now PGH2 has only 2 double bonds. But it is not over. In a way, PGH2 is like the parent prostaglandin and can divide itself and differentiates into different prostaglandin groups, such as PGE2, PGD2, and PGI2, to perform its different and specific inflammation, fever, and pain functions. Or it can also divide into thromboxane groups like TXA2, and depending on the cell's different synthase of enzyme, steps will be taken. Now going back to cyclooxygenase. As we previously mentioned, we have COX-1 and COX-2. These two variations of cyclooxygenase vary in several factors. Firstly, COX-1 is constitutive, which means it has a constant and basal production in our body, whereas COX-2 is inducible and affected by inflammation and increases drastically over prostaglandin levels. COX-1, however, has a homeostatic response, which focuses mainly on forming blood clots. Also, it is important to note that COX-1 is found only in gastric mucosa, thus in the stomach, whereas COX-2 is found throughout our body.
How does ibuprofen work? We have used ibuprofen for many situations, but not all knows how it works. Here's a quick overview on pain. When cells are damaged, they release tuning chemical called arachidonic acid. 2 enzymes named COX-1 and COX-2 then break this acid down to form prostaglandin H2, which in turn is converted to multiple chemicals, which raise body temperature, calms inflammation, and lower your body pain. Now, when certain nerve cells called nociceptors are receiving pain above this threshold, they send signals to the brain, and the brain tells something is wrong with the feeling of pain. When you feel pain, such as a cut, generally the first thing we do is searching out that bottle of ibuprofen. After swallowing 1 or 2 pills, our stomach breaks down this medicine and is absorbed into our bloodstream through the stomach wall. The ibuprofen then tracks down and attaches to 2 enzymes, named COX-1 and COX-2. This prevents the enzyme breaking down arachidonic acid. Eventually, COX-1 and COX-2 will reject the ibuprofen, and then you need to decide if you feel all better or you need to take a couple more. The effect of ibuprofen. The use of ibuprofen can cause a number of side effects like cardiovascular or circulatory events such as heart attack or stroke. The ibuprofen can also cause gastrointestinal problems such as bleeding or perforation.